what are the hard problems you bump up against and enjoy bumping up against in the biohacker, neurohacker world? What are the growth edges? I mean, I see people acquiring tools and technologies and ways of modifying their physiology and their minds constantly, and I provide some of those tools. A lot of the time we're operating in a space where we really don't fully understand what we're doing. There's somewhere between art and science.
Right.
And I'm sure half the things that biohackers do are completely irrelevant, and a certain percentage is probably working counter to what they actually want to do. What do you find the hard problems are in neurohacking or biohacking?
Mm-hmm. So neurohacking, biohacking, and what the hard problems are in medicine. And we kind of looking at that as medicine moving from dysfunction to function. Neurohacking, biohacking, generally thinking of it as trying to enhance function beyond previous baseline. We think of that as one gradient. Some of the hard problems that are not like philosophically hard problems in terms of maybe unsolvability or epistemic limits, but like the holy grails of the space. One of them has to do with complexity.
Okay.
And so when we're trying to look at what's going on for an individual person, say we're trying to look at addressing anxiety for someone, is their anxiety primarily physiogenic or psychogenic? And what causes in each of those? If we just look at physiologic causes, we see a whole good body of work on the ability for microbial imbalances in the gut to cause anxiety predispositions because of decreasing the total amount of GABA produced in the gut or serotonin produced in the gut or inflammation on the enteric nervous system or whatever, right? So if someone went traveling in the third world, got a parasite, microbiome got off, has never re-stabilized afterwards, that could be a tier 1 cause of anxiety that no amount of nootropics or CBT or DBT or anything would address, right?
Yeah.
Or if they had minor head trauma where the CAT scan or the MRI didn't show anything, but maybe a QEEG would, or maybe a SPECT scan would, because there's functional firing patterns that went off. How we're going to address that? That might require neurofeedback. That might require neurogenic chemistry, right? That's going to be totally different again. If they were exposed to mold or other forms of excitotoxins, that can cause anxiety. So there can be genetic predispositions, and then we can continue to populate. There can be methylation disorders, right? There can be thousands of different things, and then the confluence of those things, right? And then if we're looking at the psychogenic side of it, we're saying someone who had anxiety since childhood because of kind of attachment, early childhood attachment stuff with parents versus only after a very acute trauma, PTSD-like trauma. They're different psychologically, they're different neurologically, and there are different best practices and therapies to address all these things. And for one particular person's anxiety, we might be looking at some confluence of some number of these factors, right, where there was a causal cascade. And then that person with anxiety maybe also has a sleep issue or not, or maybe also has some autoimmunity. So as we're trying to help them, how do we factor all that?
Yeah, certainly.
Complexity and personalization.
Now, is this possible? I mean, I work with brains every day, and I am humbled every day by how little I actually understand. And my strategy for dealing with that is, Maslow to a man with a hammer, every problem's a nail. My hammer is neurofeedback and meditation, and to some extent nootropics. But I view the brain as a regulatory machine, and then when I do a QEEG and see something off and it matches symptoms, it's a little bit more skilled than being a mechanic, perhaps. But it's that approach. It's the let me go reach it and tweak this and see what happens. I'm working on integrating lots of different information systems so that I can, do visualization, do big data, do a better sense of what's happening. But even that will have a fraction of the degree of things you're talking about.
Right.
Is it a solvable problem? Can we actually bring in personalized medicine, if you will, personalized psychology, track data this way? I mean, is this being done? Can we do it? I think we're getting enough information about the individual data points. You can do a methylation analysis. You can do a theta-beta ratio. You can do, a blood flow imaging or something. But these measurement tools are limited by the tool space that they are using. QEEG gives me one thing.
Right.
It gives me less than half the EEG, only the dipoles that are at right angles to the skull. And it's a 10,000 foot view population average of averaged EEG across several minutes. So you're missing states completely, only emphasizing traits. And this, 30 or 40 big features in the QEEG that I look at and that I operate on in terms of intervening. But I default back to that because I can't do everything, because I can't hold everything in my mind, and I can't ask everyone all the right questions to tease out what might be going on, because I just don't understand the brain fully. I mean, as you say, probably none of us do. Are we getting to the place where we're going to be able to aggregate and analyze and extract meaning from this complexity, or is the complexity going to obscure our ability to know what's real and know what's important?
Yeah, I do believe we are, starting in the next few years.
Okay.
And having an exponential curve.
So this is a Kurzweilian prediction here?
Of sorts, in a different direction than I think much of what we hear about what is going to happen with brains from singularity.
Okay.
But as you said, we can't hold it. You can't hold it all in your brain. That's true.
Yeah.
The total amount of information exceeds our conscious processing by a lot, right?
Because of working memory limits, mostly, right?
Yeah.
There is no storage limit in the brain, as far as we can tell.
But there is a working memory limit for your ability to consciously try to make sense of not just how many different metrics, but the relationships between those metrics, right? The combinatorial relationships. So say we start looking at the genome, most things aren't affected by SNPs, right? By one SNP. There's a very few things that are. Mostly we're looking at dynamics, combinatorial dynamics across tens or hundreds of thousands
downstream
of SNPs.
Effects, essentially.
Right, but that's a lot when you look at 3 billion base pairs.
Right?
Yeah. And then we have to deal with the fact that doesn't tell us which ones are actually coding, right? We have to look at the epigenome for that, which is not something we do well right now. So we actually have to look at the transcriptome or exome, proteome, which is all stuff that is emerging but hard. And
that's just the human genes, which is not actually the majority of the genes we carry around, or actually aren't human, right? So
if you think about it this way, right? When you were saying the brain is a regulatory system, it's a top-down regulatory system, right? The bottom-up regulatory system is the code layer. The genomics, the microbiomics, the viromics, right? They are creating a distributed bottom-up regulation of individual cell function, right? And protein coding.
Sure.
That bottom-up regulatory capacity is what led to the evolution of top-down regulatory capacities that get to control larger groups of cells and tissues working together in coordination. The feedback relationships between the bottom-up and the top-down regulatory systems and all the dynamics in between is where what I think we're starting to do is map the human as a cybernetic organism, right, as a regulatory organism, apply complex adaptive system science and kind of systems modeling to understanding that better. And it is what in computer science is called an NP-complete issue. It's an uncomputable issue. There's an uncomputable amount of information. So we're not just going to brute force big data and machine learning and figure it all out.
Well, it's a chaotic system. We do not have infinite precision. Therefore, any model that steps into time far at all diverges from the chaotic system. And
so, yes, it's chaotic, meaning there's some fundamental unpredictability. There are measurement limits, and then there is uncomputable complexity. And so what that means is, rather than just depend on brute force computation, we have to understand the dynamics of causation in a complex system better so we can compute the things that actually seem like they would make sense to happen, right? That's the modeling part. I think the modeling part is key, and that's at Neurohacker what we're really focused on is taking models like cybernetics, right, like complex adaptive system science, like information theory, and putting what we understand from human biology and medicine into those more complex models to understand dynamics better, right, complex dynamics. And getting to the place where we can start to aggregate tremendous amounts of data there, and data that has progressively better sensitivity and specificity, and then be able to process that data where, right now, for the most part, when we look at a biometric, we look at someone's hemoglobin or their vitamin D or whatever, we're looking at it in relationship to a reference range.
Sure.
But when we look at someone has a kind of dynamic homeostasis, right? I actually really like your word, homeodynamics. It's a better word because the stasis is nonsense, as you mentioned.
Sorry, a knee-jerk response when I hear the word homeostasis.
Sure. But
life is not stasis.
We have these homeodynamics.
Yeah.
And then thriving health has to do with the homeodynamic capacity and resilience across all of those homeodynamic axes, right?
Mm-hmm.
Aging is decreased homeodynamic capacity across some of the axes.
And does that mean decreased range of variability, essentially?
And decreased capacity to have a stressor, right? Some things that's trying to create deviation from homeostasis and the ability to stay within the homeodynamic range.
Mm-hmm.
So then which means less resilience.
So to be concrete, things like insulin or cortisol, let's say insulin rises when you consume sugar.
Right.
When, it's a signaling molecule, cells suck up the glucose, insulin goes back down. If you chronically perturb the insulin system to the top of its range, eventually it stops varying.
And we call that type 2 diabetes. And on the way there, the kind of syndrome X dynamics where fasting insulin levels raise, you get insulin resistance at the level of cells. Now you take sugar in and your blood sugar is going to go further out of balance for longer, right? Causing more effect on other systems that are affected by blood sugar, so more than pathophysiology. So we're defining here, aging is not disease, because disease we'd actually say is the deviation from homeostatic range or homeodynamic range, where some metric actually goes out of effective range, and then you get a pathophysiologic cascade, right?
Yep.
So aging is not that, it's the increased susceptibility for that. So it's increased susceptibility for disease, because someone could have all of their markers within homeostatic state range, but more sensitive to them being able to go out of range from whatever stressors.
So they're less labeled to sit in the healthy basin of the phase space, so to speak.
Right.
Okay. All right.
And so what we're interested in here is how do we, where someone already has disease dynamics going on, how do we identify specifically what caused deviation from homeostasis on which dynamics, what was the causal cascade that happened, and how do we specifically reverse it? And like we mentioned with the anxiety, are we chelating neurotoxins, or are we fixing the gut-brain axis, or those are very different things. So it requires specific addressing of what the causal cascade was.
So this is not, let's say, go back to insulin, the insulin insensitivity that cells experience after high levels of insulin, that's not the disease itself. That is the failure of the dynamic range.
Correct.
And then we call these diseases things like diabetes and Alzheimer's and other things because of the failure.
Now, there are some people who get type 2 diabetes who did not consume too much carbohydrates relative to what they burn, but they are deficient in chromium or vanadium that are necessary to actually have insulin process at receptors. So there are other dynamics that can go on, which is why we actually have to understand what is the entire causal phase space of what can cause an imbalance, all the things that could be involved. And this is what we've sucked at medicine so far, is dealing with complex causation.
Yeah.
Acute causation, straightforward, right? So injury, pretty straightforward, and so we're going to be able to address that in a clear way. But where the causation is either delayed in time, right? So something happened and it led to a slow deviation from homeodynamics over the course of years or decades, it's hard to assess what it was.
Yeah.
Or where it's multifactorial. There's many different things going on, and where it's a combination of some multifactorial set that's different for different people. So I don't think MS is a disease. I think it is, and I don't think Alzheimer's is a disease or anxiety. I think they are conditions where some cluster of symptoms and biomarkers come together where we call it that, but the actual causal etiology going into it can be radically different from one person to the next.
So more of a syndrome, less of a disease, a cluster of symptoms that may have multiple causal features. Interesting. I saw a study out of UCLA a couple weeks ago that suggests Alzheimer's insulin insensitivity, if you will, can be reduced simply through behavioral mechanisms. They took a bunch of people off of all the meds, off of anything, and instead of doing a specific intervention, they said, let's do lifestyle things. And did, paleo diets and exercise, and lo and behold, symptomatic Alzheimer's individuals showed recovery of some function.
Totally.
This is fairly magical things. There was a study out a couple weeks ago showing that even in people with type 2 and type 1 diabetes who have no beta islet cells essentially anymore in their pancreas producing insulin, short period of fasting, I forget, was it 12 or 24 hours, a very short period, seems to cause a redifferentiation of non-beta islet cells into beta islet cells to produce insulin.
Right.
That flies in the face of what we thought of in the disease model of diabetes, where it's either autoimmune or it's toxic from the sugar essentially that kills insulin-producing cells. Once they're gone, they're gone. That does not appear to be accurate. Is this the kind of thing that you're digging into?
It's the same thing with the idea that we don't produce new neurons, right? That was an old idea.
It's true.
And then we realized neurogenesis happens and it can be upregulated.
Yeah, and then it was only in 3 places, and now it's pretty much everywhere we look in the brain, we're finding new pluripotent stem cells.
And so can we increase pluripotent stem cell production and their differentiation throughout the connectome? And can we increase neuroprotection and synaptogenesis? And like we're learning a lot about how that works. When you have a system that generated itself, it was a self-generating system, the idea that it just could not generate those tissues that it originally generated, again, doesn't make that much sense. There was obviously a capacity to do it, and then the capacity decreases or turns off for some reasons. Those then likely are modulatable to turn on again. So when we think about life extension, how do we increase progenitor stem cells? How do we increase the rate of purging senescent structures? How do we protect cells from senescence? These are all things that happen already, but they can start happening at suboptimal levels for a lot of reasons. We can support them happening at optimal levels and maybe even beyond previous genetic optimality.
Right. Just to give us a discrete example, I'm a gerontologist, teach courses in aging. There's a term in aging called antagonistic pleiotropy. The idea that a mechanism that is healthy at one point in life is harmful at another point in life. And the telomeres, the shortening end caps that prevent replication forever, when you're young, prevents cancer from running away with your cells.
Right.
When you're old, keeps fibroblasts and your collagen from cleaning themselves up, and they become homes for a pro-cancer environment, basically. So it's this kind of thing where you're identifying where the natural arc of development may not be optimal for long-term performance and turning these things back on and controlling it. I mean, from the point of view of telomeres, for a long time that was, of course, the holy grail in antisenescence, and now we realize if we could give everyone a pill to lengthen telomeres, ridiculously, it would probably just cause huge amounts of cancer. So we can't simply reach in and turn one thing on. I mean, this is something I struggle with in neurofeedback. It's not reaching in and dialing up and down different neurotransmitters or different, brainwave ratios. When I perturb a system, the system reorganizes, and I'm never quite sure how it's going to reorganize. And this is, a little frustrating sometimes.
Well, so if we think of it as increasing system resilience, right, homeodynamic capacity, because it is a self-organizing system and the complexity is well beyond what we're going to understand in a momentary view, right? We're never going to factor all of the dynamics happening, probably even in one cell. But we can understand the dynamics that are involved in some of the key ones in its own regulatory dynamics and how we can increase its regulatory capacity. And so in general, do we see things like redox signaling decreasing in efficiency in cells? Yes, right? So this is why all the nicotinamide riboside stuff has been so exciting. Is if you can upregulate the NAD plus NADH ratio, there's a lot of cool things that happen. Do we see ATP decreasing inside of cells? Yes. Do all the things that cells do need those chemicals? Sure. So if we can support mitochondrial biogenesis, if we can support ATP production efficiency and Krebs cycle upregulation, if we can support protection of the mitochondria so they don't become senescent, right? These are key things that, what exactly is that going to do? Well, it's going to help the complex dynamics of everything that the cells are doing happen better. And so I think those are one of the key things we were saying earlier, there are specific sources of pathology, right? Causes of deviation from homeostasis that we want to identify specifically and reverse. Pathogens, toxins, deficiencies, etc. Then there are general system dynamics that support system resilience, and we want to support those even though the effects of that are going to be general, increased well-being across systems. So EEG regulation is going to produce, how much is it going to affect a particular gastrointestinal dynamic. Well, it depends on what all the causal dynamics are, but it should move it in the right direction to the degree that it's creating better sympathetic, parasympathetic, ratio dynamics, etc.
Yeah.
So we want to, in general, support the homeostatic processes that are fundamental and get better at diagnosing individual sources of pathology and reversing them.
This is great. I mean, this actually ties into how I often frame neurofeedback for people, and they say, oh, I want to do this. And I'll say, well, we're probably going to give you more resources in all of the regulatory domains of sleep, stress, and attention. And what that means is resilience.
Right.
And we can probably reach in and do more specific things for you. But let's see what happens first when we bring the resilience way up and get you sleeping like a rock at will and being creative and flexible. Let's do that for the first few weeks. And then we can go in and try to target the thing you think you care about.
Right.
And usually the thing that they really want to work on sorts itself out along the way towards just building everything.
The complexity of the interconnectedness of everything is, again, why specialties have failed at complex illnesses.
Yeah.
But it's also so fascinating because, we can look at a structural issue, right? Like a postural issue or osteoarthritis that's causing continuous inflammation, where then the inflammatory molecules, right, cytokines, can cross the blood-brain barrier, cause neural inflammation and damage to neural circuits.
Yep.
Also, where the continuous pain dynamic is causing sympathetic feedback into the brain, which then is going to cause effects on all the things the brain is regulating from a top-down perspective.
Right.
And what needed fixed was a structural issue in a knee.
Right.
Right? Or mold in the environment, or a runaway process of dysfunctional information processing in the brain from a particular psychologic meaning-making pattern, right? So can structural issues in joints, or psychological patterns of focus and meaning-making, or things going on in the gut, or affect any other system in the body? Yes. This is what's fascinating, and it's why trying to separate neurology and gastroenterology and oncology doesn't
futile,
work. Right? And so now it's how do we synthesize what we understand about the parts to understand what happens for whole systems and what happens across systems.
So now you're doing this at the Neurohack Collective with this idea of bringing in different aggregate data types and modeling and trying to figure out what is the meaningful inflection points for these models where, where's change happening. That's very, high level. And those are the kind of projects that frustrate PhD students for, 20, 30 years.
Yeah.